Okay, we're going to have all the speakers come up on stage for the panel discussion. So anyone who's still here, the room is still very full, so I'm so happy. I'm so happy. It's a long day, but you guys were practically on time. I knew Global was going to go over a lot, but they were so amazing, and they've done so much, and they're so proud of themselves, and we're so proud of them. So come on up if all the speakers. Ellen, do you want to come as well and represent Global in case there are questions? Everyone, yeah. So now's the time. Get creative. Think about your questions. You all ask a lot of questions on Facebook, but you don't get the researchers answering them. So now's the time for you all to ask them directly your questions and get answers from the horse's mouth. So we got a couple questions earlier in the week. We had recruited for questions online. So I have a few questions, but first I want to give an opportunity to those in the room who want to directly ask questions to all of our speakers today. And I'm sure Ed has lots of questions for everyone as well, being the real neuroscientist standing on the floor. Any questions in the room? Stand up if you have a question. Okay, runners, get to it.
Coming. Here.
Hello. I have a question for Nikki. And I was wondering which is the estimated undiagnosed Angelman syndrome population?
Yeah, that's a really great question, and I'm not sure that anyone knows the answer to that. I think it's going to vary greatly based on the age of the particular individual or the kind of the birth, what point their birth happened. It's also going to vary quite a bit throughout the world. Some countries have more readily available genetic testing than others. I'm not sure that anyone knows a good estimate of that, but if someone else on the panel has seen a good number of how many folks we've missed, I think it's a huge number.
It's huge. I mean, the incidence is estimated to potentially have 500,000 people in the world living with Angelman, and we probably know and have identified close to 3,000 between FAST and ASF. And I would say that we were told ICD-10 codes in the US, people like industry that has gone in and looked at ICD-10 codes for Angelman may have documented about 7,500. It means just in our country, we have not identified those that have a diagnosis of Angelman syndrome. So it's enormous, unfortunately.
Hi, my name's Michelle. I'm from Australia. I work in the technology company, and as you know, at the moment, machine learning has been around for decades now. We are entering into the stage of Gen AI. So my question to everyone is, how does Gen AI play into the roadmap in terms of therapeutics, but also data collection and test cases for using said data? So whether we're going to create a data lake which can be accessible for Gen AI tools such as Power Automate to develop, papers or policy changes, or from a clinical and research perspective, whether they can identify, patterns based on the experiments that you conduct.
On how you're using AI?
With Ladder, we are currently working with our data science folks at RTI to try to develop a prototype for an AI-powered chatbot to mine the data in Ladder to answer community questions. We're very, very early in that process, but we are looking into that.
Maybe you can comment a little bit about off-target analysis genetically, because Youngwei, for off-target analysis, you're using some machine learning, right, to look at off-targets for gene editing for different genes. So maybe you can comment a little bit about how you're using some artificial intelligence and machine learning when we look at off-target analysis and bioinformatics and predictions, which would be considered machine learning.
Yeah, I guess it's still kind of work in progress almost for every project. Clearly, we will do a lot of sort of bioinformatics, machine learning first. Right now, AI has been incorporated clinical diagnosis and analyze the genome exon. I'm sure that's will be the part of this ongoing process use the AI, whether it's the question oftentimes is how AI, you need some kind of machine learning, you need a larger data offset, off-target evaluation data set. Right now, it's all individual lab. There's no shared resource right now. We actually, in the consortium, we discussed whether each individual side doing the off-target analysis and which platform, which cell line, which cell type can all share data together. So now you can have a large data set and allow to making better prediction for off-target events. So.
Yeah.
Maybe I can make one comment. I received an email from somebody who wants to use AI to advance Angelman research. I encourage that person to come to this meeting, and I assume maybe they're not here, or they should stand up and identify themselves. But there is, I can share, I think, the contact for this person.
Hey, it's Terry Jo Bichelle from Combined Brain. It's really amazing to see all you sitting up there. It just is so inspiring, and all the stuff that's happening is just fantastic, and the fact that everyone's building off everyone else. But my question is to Ryan Fisher. Angelman is ahead of a lot of other disorders, but we still have a lot to learn from, everybody else's successes and mistakes. And Ryan's been around the block with a couple of other disorders, and I'd like to ask him, what's the one thing that you learned from Duchenne and your experience there that you really wish Angelman would jump on?
Well, I think in some ways we're doing it because in Duchenne, I don't think the patient population was engaged early enough. Even in interpretation of outcome measures and sort of lending context to how meaningful they may be. I'll give you an example. In the 6-minute walk test that was used in Duchenne, no one actually asked the patient population, do you care about this? And it was used over and over again in trials, and then having to explain how is a 30-meter difference meaningful to the population was a challenge. I think through ABOM and through their surveying efforts, we're getting a bit ahead of this, which is a good thing. And I think that's one of our biggest lessons from what we did earlier. And keep in mind, we now have legislation that's mandating that the FDA incorporate the views of patients and caregivers into the process. When we started, we didn't really have that. So I wish we would have done more through outcome measures, through the development of what we're doing in ABOM earlier in Duchenne, because I'm seeing a lot of really good progress because of that in Angelman syndrome.
One question here.
Hi, Emma again from the UK. Firstly, I just wanted to say thank you. What an amazing day. My brain hurts from all the amazing information. And for those watching it online, I've done it for a few years watching from home, and it doesn't have the same impact. So do come over next year. It's amazing. My question, we've talked a lot today about all of the different pipelines for the different pillars. Alison, you said for every 10 drugs that make it into clinical trial, only one gets through the final hurdle, and we've talked about sort of the pillar 3 therapeutics, etc. So my question is 2 or 3-parter really, but what happens to the children that are on the trial if we're pushing all the horses forward, but the drug that the child's on in the trial isn't the best horse? Can they switch at a later date? And then if you combine some of the therapeutics for like we've got it written down, the BDNF, the SYN3, for example, with Ionis or Ultragenics, how does that work? And then also for parents that are still thinking about, do I put my child on a trial or not? How do they know whether to wait for something in the future or make that decision now?
There are a lot of layers to those questions. And tomorrow we're going to be talking a lot more about clinical trials, and I think that you're going to get a lot of these questions probably not answered, but you're going to feel more comfortable with understanding how to make those decisions. And I can just briefly try to cover some of them. I forget them all, but I can briefly try to cover some of them because I think there's not clinicians on the floor or clinical trialists on the floor that can probably do that. But so I'll do my best just from the perspective of drug development. And what I would say is that in a clinical trial, it is very unlikely that you're going to be able to do 2 drugs at the same time when neither one of them are approved, right? So until there's approvals, you're not going to be able to add one drug to another drug. So that's firstly. Secondly, it really is going to depend on the modality. So, for example, when Ovid had their clinical trial and it failed, and people were on that small molecule, and washout is in 8 hours. The half-life is 8 hours. So washout is a couple weeks at best. You're going to be able to get off a drug, wash out, and there's probably not going to be an interaction of that drug with a gene therapy or an ASO. So a small molecule, a downstream target, something that needs replicated dosing that's not gene targeted, is probably going to have a different risk profile of what that washout period would be and who could be included in another trial. If someone gets an ASO, it is, and we'll talk about this tomorrow when we'll let the sponsors answer this question, it's very unlikely that they're going to be included in another clinical trial that's gene targeted or gene modifying, because no one really understands yet the impact that first drug that modified the gene in any way could have on their drug. And they need to understand the safety and the efficacy of their drug in a naive state. So it's going to be very hard to combine them unless one of them is approved and becomes standard of care, in which case there could be a really hard time recruiting, and you're going to have to look at things in combination. And so until there's many approved therapies and then we can look at them in combination post-market, in the clinical trial space, that's going to be very hard. Making those decisions on, do I do a one-time therapy in which I can never go back versus a repeat therapy and we can see the effect and come off of it if we don't like it, that is going to be a very personal decision, and that's going to be a very complicated decision. And it's going to be different for different families that have different experiences, are from different parts of the world, and have children that are willing or not willing to go to a hospital frequently or unfrequently. And so I think that there's going to be enough people with different preferences of what they really want. We did a patient preference study just looking at gene therapy, a real small one. We did a little round table on this. And what we realized was certain people were all about gene therapy. They wanted a one and done. They don't want to go back to the hospital multiple times. Some were really excited about ex vivo bone marrow transplant, keep it pumping out of my bone marrow all the time. And others were like, I don't know about that. I think I just want it contained in the brain, and I'm more comfortable with a brain delivery. Others were like, I don't want to be in a phase 1/2, but I'd be comfortable with a phase 3 once I see the safety of the phase 1/2. Others were like, I want to be first online. I want to get it first because if I have to wait 3 years for the phase 1/2 to finish, I don't want to wait 3 years to go into the phase 3 where I might be in a sham or a placebo control. So everyone just looks at this very differently, and it really is going to require a lot of counseling with your provider and the PI at the site running these trials to talk you through the process, talk you through the risk and the benefit. It's nothing we're going to be able to tell you because it's going to be so incredibly personal. But it's a big decision, and it's going to be a calculated decision that we're going to help give you the knowledge and the power to make those decisions and ask those questions because they're very important. So I didn't answer your questions, but I told you it's hard.
One question here.
My son, if a person, one of the children or young boys or girls have mutation, only mutation, but not the deletion, the investigation is the same, or is a shorter investigation if it's only a mutation, or it's different investigation, no? Could be a more longer the investigation of a deletion than a mutation, or it's the same thing?
As of now, it's the same in terms of what the opportunities are and what the length and the duration of any of the investigations. But we're going to talk about clinical trials tomorrow, and different companies are working with deletion only or deletion and mutation or UPD-ICD, and eventually all of them. So we'll answer a lot more of that tomorrow, but where it stands right now is all of the mouse models that you saw today, everyone uses essentially the same model, Dr. Zhang's model. Everyone uses the same exact model. That's really a mutation model. It's a deletion within the gene, so it looks like a clinical mutation. And so really all the testing has been done in a mutation model, not in a deletion model. We're working now to do the deletion model so we can understand the genes outside of UBE3A. So really, everything going into clinical trials based on this preclinical work is based on the mutation. But the deletion we know is missing UBE3A, therefore it applies to deletion as well. The UPD and the ICD it also really applies to too. But remember, paternal activation of those genotypes means you're unsilencing 2 alleles and not 1 allele. And that's where the trepidation historically has been, is let's understand this in mutation deletion first, and then we're going to understand paternal unsilencing in UPD and ICD as well.
Coming over.
Hi, a couple of questions about UPD, and I'm not sure if anybody knows anything about this, but has anything come out in the last year? There's been discussion about for paternal activation whether UPD ICD would require half dosing or double the dosing. Has anything new come out to understand whether or not you think either one of those is right?
To Art and Young Wei, because the geneticists in the room should talk about that.
I don't think we know at the time, and it may be that certain correction mechanisms where it wouldn't matter. I mean, if you did gene therapy, it would, the fact that it was UPD wouldn't be a factor. So it may be different for the different therapies. I do think those patients do stand to benefit, and there is some risk of overexpression, but I am anxious to see them included in trials. So we get, the only way we're going to know the answer is by treating UPD patients and seeing what happens.
And the only published data that's really out there is from Stormy Chamberlain, and actually maybe Eric can comment on this. She showed UPD, iPSC cell lines and neurons showing they needed half the dose. That's the only data that's out there, is that they responded similarly to mutation, or I think it was deletion, the comparator, and it was half the dose in cell lines that responded the same. But that's all we know.
Thank you. One other question about UPD. There has been research that's come out in the last couple of years that has kind of ranked, if you will, the abilities of the different genotypes, putting UPD at the bottom, for lack of a better term, of the non-deletion genotypes, which makes me believe that something else is going on with the genes from the paternal allele that are double expressing when in neurotypicals they are not. Is there any research? I know that Albert Kyung was doing some with the landing pads, but has there been any research or any suggested research to identify, just like we're doing with the other drugs on the deletion, on other genes on the deletion, what else is happening with UPD aside from UBE3A?
No, we haven't.
Who wants to look at that?
Have you looked at that? Because I know we've talked about it many times.
We
and I
do.
don't know, Albert's in the room, he may be able to comment as well.
As a part of it, we generate a larger deletion for 6 megabase. We indeed generate also duplication of the same region, but we have not, we just focus on the maternal duplication right now, but it definitely has opportunity to look at the paternal duplication, see if anything will pop out from there. Molecularly, I mean, indeed, the 2 dose is a question. Not every gene is dosage sensitive, for sure, and it's very hard to oftentimes assess the. So the when miss it's easy, and when the actual dose and for determine whether it's truly dosage sensitive, and using behavior analysis oftentimes it's also not very sensitive either. We only look at one snapshot. But it's clear we have a material right now, part of a FAST support. We made initial only making this deletion mice or the UB3 maintain, delete everything else. Part shall not do. She's keep explore all this opportunity, and she generate many other line of mice. We just don't have time right now to look at every line very detailed right now. But definitely it's in our plan. So
I think the question alluded to the potential problem that all of the paternally expressed genes are being overexpressed. And so there's a theoretical, but I'm not aware that anybody has any data about that and whether it causes any harm or whatever. But I think there are going to be variations from patient to patient that we just don't predict. I think one of the most striking so far is that patients with mutations who I would have thought would be the very most suited for benefit, that some of these mutations are dominant negative and they actually don't respond. So who's going to respond and who isn't going to respond is going to vary a lot from patient to patient, I think, depending on their ages and various kinds of things. But I think it is interesting. Does anybody know anything about overexpression of the paternal genes in UPD?
Yeah, so it's a multiple different model, just like Banfield Pod and Matt Anderson generated as a sort of transgenic only UP3A coding region has multiple copy. So that data, I would say, is a little bit inconsistent. Clearly, they focus on most of the maternal again. Even maternal, the actual duplication, one duplication, almost not many phenotype in the mice. So now when you duplicate it 2 or 3 times, now you see some phenotype. So therefore, that kind of suggesting maybe just 2 copy at least for mice, it's not significant impact at all. But it's clearly, because maternal you have not seen anything, but there is one model, interestingly, in the mice, and the Japanese group made a long time ago. They somehow see opposite. Paternal duplication in this 15q11, q13 region in the mice has phenotype, but maternal has no phenotype in the mice. So it's still very, very peculiar why in that particular line. So we actually have a new line, and our mice with maternal duplication is different from that line. So it's maybe the design for how the mice was generated at that time. So it's a lot to still to be learned for sure.
Yeah, I just want to add to that by saying that I think a lot of this discussion is kind of presupposing that if we could bring UB3A up to neurotypical levels in a UPD patient, would they have kind of a double, right? But I think it's equally likely at this point that we might not hit all the neurons and everything. We might only get it up to maybe 50% of neurotypical levels. And if you remember Allison's remarks earlier in the day talking about chimerics, even that might look like a perfect rescue, even though it's only expressing half of the neurotypical level. So in that case, if you had UPD, you would still be in a range where even if you had double the expression of someone that was treated, it would still be just getting up to the neurotypical level. So, that's even considering other kinds of things. I think that Dr. Baudet was trying to, was mentioning. I mean, there's some evidence that UB3A is activity dependent. So if the neurons are active, there might be a higher level, there might be a lower level if they're not active. There could be autoregulatory functions that we don't really understand at this point. So I mean, there's a lot of things that would go into what would happen with a UPD patient than just strictly looking at the genotype and saying, well, it's going to be double.
I think ultimately that's not Lauren's question about UPD overexpression. Her bigger question is about the SNO RNAs and the other genes in the UPD ICD genotype where they're also overexpressed. If they're naturally silenced, they're overexpressed. So if you use a paternal activation strategy, would you then repress those other overexpressed genes, in which case you wouldn't have overexpression of the SNO RNAs and the genes outside of UBE3A. But her question was, we're looking at all the genes around UBE3A for deletion. We're not looking at the genes outside of UBE3A for UPD ICD, where we have an underexpression problem in deletion. They have an overexpression problem for the paternally expressed genes that are expressing with the silencing. So that's what she's trying to understand is, are we looking at those?
And it comes from a relatively recent study that ranked, for lack of a better term, the cognition and the clinical, and Dr. Sadwani might be able to speak to this, but the clinical presentations of each of the genotypes separately. And while non-deletion is admittedly ahead of deletion, UPD is 3 of 4. So something else has to be going on, because mutation and ICD do better. So it just logically to me makes me feel like something besides UBE3A is happening in UPD, if that makes sense.
I think that one point that should be clear, there's talk about the other genes in deletions, and those are mostly genes that are not subject to imprinting, and those would be not a problem in UPD. I think the biggest potential problem in UPD is that overexpression of SNRPN or SNO RNAs is harmful.
They are, and if they are do we have any strategies to think about how we're going to deal with that for UPD?
I'm not aware of any published data.
I'm not aware of any either, though I know that we kind of need to fix the obvious problem before we can solve the rest of it. I also think we should be cognizant of the fact, I know you talk about this a lot, Lauren, of this graph, but remember that 3% of the population of a small population in that study was UPD, and UPD and ICD really should behave the same genetically, and they spread out a little bit, but
they do.
It is magnified when you look at that compared to the neurotypical population. So I think part of this could also be sample and error. So we don't want to I know you like to get hung up on this, but I don't know that you should yet, but it doesn't mean we're not looking at it and we're not thinking about it. But I do think that it is something that may not be as dramatic, hopefully, as in your mind it is, because I know you can't get that graph out of your head because we've talked about this for years. Anjali, I think it was your paper actually that showed the difference of UPD to mutation.
Yeah, I think, just clinically seeing these kids, I wonder, sometimes there's an underestimation in our UPD kids just because they have more behavioral challenges. So I'm wondering whether they're not able to show their true potential because the behavior is somewhat confabulating that situation. So, clinically, I'm not necessarily seeing the difference. Yes, our paper showed it, but I do wonder whether the behavior challenges is underestimating their skills.
So Lauren, just to add to this from a molecular standpoint, we did have a nice presentation on the biorepository of the different stem cells, and I think one of the big advantages of that is that it's going to have all the different genotypes, and the amount of studies that you could do to help sort of start teasing some of this out could be really interesting. So I think we're going to get there and start understanding some deeper layers.
Any other questions? I had a couple online. Ed, if you have any, go for it while I look at these.
So I got this question from a couple different parents, and I think that this could be directed to anybody. Maybe Dr. Marshall would want to answer this. Why there's not more potential therapeutics in Pillar 3? So they were saying there's a lot in Pillar 1, even more in Pillar 2. Why are there not more in Pillar 3? Why do you think that is?
I think we need more basic research to understand exactly what UB3A is doing and the mechanisms of what is affecting. I mean, we're just starting to understand that it regulates autophagy, but we don't exactly know how it's doing that. That's where Dr. Mayer's work is so important in identifying the actual substrates, because there are a lot of substrates out there. I don't believe they're real substrates. I think we need to identify the actual substrates, and that will give us insights into exactly what UB3A is doing. As I mentioned, it's also involved in transcription. And also when you look at ubiquination, autophagy involves ubiquination. So when you knock down UB3A, you're going to see increased autophagy, you're going to see ubiquination of all sorts of things that is a general thing. So it's not that informative. So obviously we're focusing on a treatment. That's the key thing. But because, my lab's been doing all this basic research, that's why we've been able to develop something in Pillar 3 based on the basic research and understanding the role of this target, PS95, in TrkB signaling. And I've been working on trafficking of glutamate receptors for 20 years, so I've only come into the field very recently. So, yeah, it's just a lack of basic understanding of what UB3A does. And as we learn more, Dr. Wong was talking about autophagy, drugs to treat autophagy. That could be another approach. And then I know Dr. Wong has a paper submitted looking at estrogen drugs. So as we understand more, we're going to identify more drugs. And then, obviously Ben Philpott, had that paper in Nature Communications where they identified a small molecule. It's very exciting that it could actually turn on paternal expression. We don't understand how it works. I mean, that would be great to understand exactly how it works, but that could be the tip of the iceberg. If we can identify a chemical, a small molecule that can taken orally, that can turn on the paternal gene, that's a huge breakthrough. I mean, Ben did not have any data on behavior in that paper. Maybe that's coming. We have to wait and see, but that's incredibly exciting. A whole new approach, to treating this disorder.
And maybe exemplifies why we need to continue basic science research, right? There's still a lot that's just not known.
We are not there yet. Okay, well, we're at overtime, so I'm going to...
Can I make one?
You can, because I'm going to make everybody make one final comment.
Oh,
but you can make your comment first, because then I'm going to make you make a different final comment.
My comment is, in the session that immediately preceded this one, I thought it was striking that there was no one from China, no one from India, no one from Russia, no one from Indonesia, and no one from Africa. Those include, Indonesia, China, and India have huge populations, and maybe there were a few dots on one map there, but I think trying to get somebody from those countries here to participate in this event in a year would be really exciting.
You have people from China and Hong Kong, raise your hand, shout it out, in the room. And I went to China this year and met with the Chinese Angelman Foundation, thanks to Yang Wei, who helped establish that foundation. And they have 1,500 families that see one clinic in China. And so it's enormous, the population, and they are very separated from everything outside of China. And I think that's unfortunately part of the Chinese culture in that regard. But Yang Wei is working really hard to try to get the Chinese community to join our community. So we're actively working at it, and Ellen is working hard with India and others as well. But yes, it's very important. It's very important. So as we end the day, I do this every year, so I'm going to do it again. So get ready. I'm going to ask all of the researchers, I'm going to ask all of the people that do not live with a child with Angelman syndrome, because it's going to be too hard for you to do this. And you want to hear it from everybody else. I want each of you to just tell our community, so we all are here looking for hope. And you guys showed us science, and science to me is the epitome of hope. When you see real science become a reality, that's when hope becomes real. And so you're working really, really hard for our kids, and you don't have a kid that lives with Angelman syndrome. I have to work hard for this. I don't have a choice, but you do. If you can give 2 sentences to the community related to the work you've done and seen, it doesn't have to be hopeful, I suspect it will be, that you feel the community should hear. One take-home message from the work you do and what you've seen today and what you see in the field. Give them a message, 2 sentences each. Zhang Bing, go.
Sure. Relevant to Angelman syndrome, we mainly work on genome editing. So as I mentioned, this is a relatively new technology. The first article about application of this technology for mammalian cells was published in 2006. 13. It's just 10 years past. Probably some of you know that the first CRISPR drug was approved last year. Is a one per lifetime treatment. So I think now we really at the time, probably we are positioned to do that, to replicate the success for Angelman patients and family. Hopefully we can get there in the next few years. Thanks.
So far to me. As some of you may know, I was not trained in genetics. I changed my major from plastic surgery since I somehow, many times, I have the thought that I was raised up by FAST Foundation. I started my basic science research with Dr. Jiang, and the first project I work out is on the gene therapy for the FAST. And I just feel this is really the work I like to pursue for my entire career. So I feel since I was raised up by this foundation, I really love to consider every children as my sister brothers, if you don't mind. You are my family, and you give me career. I hope I can do my best to give a new life for you.
So when I listen to Dr. Bodell walking through the history, I probably have a very different reflection than every one of you. So clearly when I started this project, then I loved his lab. For all different reasons, I was not, the Angelman project was a low profile in my lab because for all the practical reason, funding reason. But then Allison found me and in the middle said, I need to kind of raise the profile again. So now we end up with this very successful project. So I really think the community has helped my career a lot, and I really hope I can pay back for all the support so many years. The community support me, and the hope is clearly much higher than when I started. And every year coming, is hope is even higher every year I look at it.
I've been studying the developmental trajectory of your children with Angelman syndrome for the last 14 years. I am so excited to be part of this revolution which is happening right now with the upcoming clinical trials and the ones in the future, because I truly believe we have the potential to change this trajectory. I cannot wait to be part of this, so thank you for letting me be here.
So it's been an honor to serve as the SAB chair, and what I would say is it gives me a unique perspective and respect for this community, because you should be so proud about the variety of therapeutics that you are able to support and embrace, because that is where the strength is. We are talking not just about one therapy, we're actually talking about layering them on top of each other, and that's amazing. And the other idea is just FAST will take chances. They don't say you need to have 50% of this study done before we want to fund it. If you have an amazing and great idea, FAST will take that chance. And that is why there is the variety of potential treatments.
I spoke earlier, but I would have one comment. It's really been amazing to me. Most of us, most clinicians and scientists 20 years ago thought these disorders would be untreatable forever, and were relatively hopeless about the situation. Obviously, it's a lot more positive now. But it just takes hard work, and I think cystic fibrosis has a good example. That was a disorder where I would have bet everything that gene therapy was the solution, and it's been small molecules that have changed the life for cystic fibrosis patients. Very hard to predict, and I think also in the case of cystic fibrosis, there was an aggressive organization of parents pushing the situation, much like here, where they founded a company and so on. So I think FAST is having a big impact on this. It wouldn't be where it is now without FAST.
So I'll probably sound like a broken record, but your voice is your power. And I really encourage you to get active in various ways with advocacy, with data collection, whether it's natural history, whether that's surveys. And remember, this is informing so many people who don't live or understand the lived experience of Angelman syndrome. So if we have more people that are understanding that, they're going to make better decisions on behalf of you and your loved ones. So your voice is your power. Always remember that.
Is this on? Yeah, okay. I would say, so I have had the privilege of working with multiple different rare disease communities and the honor to work with both FAST and the ASF. And I would say that in my experiences in the last decade working in Angelman syndrome, this is the most resilient and passionate community that I've had experience with. And I just, from that, what you all have done, what I've witnessed you all as a community of families do to move treatment forward for your children is just so remarkable. And it just seems to me that when you all are working together, when we're all working together as a community, there's really nothing we can't get to. And so it really lends a lot of optimism. So I'm grateful to be part of it.
I would add to what Anne just said about passionate and resilient, also thoughtful and thorough. I've also worked with a lot of different rare diseases throughout my career, and what has already been established is more advanced in other conditions that I've worked in that had approved therapies. I mean, that just the amount of attention and time that it's been spent to figure out everything that's going to be needed to get therapies through the door is amazing and incredible. So it's an amazing world to be in.
I hope to continue our own work and establish new collaborations to really understand the contributing role of the non-UBE3A genes and hopefully establish that as a pillar for new therapeutic development to complement all the other exciting work that we've heard about.
Megan, you have to change the graphic. We're 5 pillars now.
No. I hate giving talking about this, but I have to say that I don't think we have to dream or hope anymore. I think we're on the cusp of major breakthroughs. We're seeing that, and hopefully with the clinical trial data, we'll show that these new treatments are working. Also, I don't really believe in AI. I'm a great believer in HI, human intelligence, and I think Allison has brought in the best minds in the field to solve these problems, and that's why we're at this point, at this time. Thank you.
I think my answer would be two-pronged. One related to my research, and so with the gene therapy, why I'm so hopeful. But I think it's following off of Dr. Marshall's idea that we don't need to have hope, we can have confidence in our results. Because it may seem like my therapy is brand new, revolutionary, never been done before, but it has been done before with other diseases. So we're following a roadmap that we know is successful. There are patients who had diseases who have been treated with gene therapy that are walking around today happy, healthy, completely cured of this disease. So we're not trying to do something that's never been done before. We're doing something that has been done before with a different disease. So confidence that it will work, I don't think we need to have hope. I think we can be confident that it will work. And then, as in general, with the field of Angelman research, I think I think it's been kind of reiterating other people's points, but the collaboration is really, really awesome in this community. So, I'm an immunologist by trade. I'm not a neurobiologist, but working with Allison and the FAST team, they've connected me to so many people who can help fill those gaps. You don't need to know everything. There are sort of experts everywhere, and so being able to work together, knowing what you know, but also knowing what you don't know, and having confidence, and being okay knowing that I don't know everything, but there's someone in this FAST community who can help me learn what I don't know, has been really encouraging.
So, a while ago, Ed Weber convinced me to work on a gene therapy for Angelman syndrome, and it's kind of interesting looking back. there were no gene therapies at that time. There were no approved gene therapies when I got started on this. And, you know, I didn't know anything about like clinical outcome measures. I didn't even know that was a thing. And, you know, certainly we tried to put together a program that we thought would advance toward the clinic, but, you know, I don't think we had a really clear idea at that time. And, you know, we didn't have anything to point to. Now there's like 30 like cell and gene therapies that are approved. There's like 100 more coming. And so now I'm a believer, and you should be too.
And I think we're going to close it out. Ed, I'd like you to answer that question as someone who's been with us from the beginning, well before my time, from academia to industry to the community.
Yeah, well, thank you. So those of you who don't know, I went from academia, 20 years in the laboratory, running my own lab, and working in AS to going into industry. I was in industry for 5 years. When you're in industry, you're kind of out of it. You know, I was outside of the community, outside of what was going on, very focused on a lot of the things that were going on with the company. And now 5 years later, I come up kind of like, you know, come up for air and look around. You know, you sit at a conference and you, excuse my language, have, you know, a holy crap moment. And I had like 5 of them today, and I know the science. And so when I see this stuff on the screen, I have enough knowledge to understand what it means. And I hope that you kind of got that from the people that are up here talking about it. There's a lot going on. And when you say you're on the cusp of really great things, I mean, look at the research that's happening. Something that we never dreamed about when we started FIRE. We were still trying to understand, you know, what UBE3A was doing and how we could identify those targets and where we could possibly go. And now there's things that we had never even dreamed of. You know, CRISPR came online, HSC. There's so many different types of research that are going on that's just flying toward potential clinical trial. It's really exciting, and I'm just happy to be part of it again. And thank you for allowing me to be there. Thank you.
So thank you everyone for sticking it out. I do that every year. But I think it's important that you hear from them, because Ed and I hear it every day. We talk to these guys all the time, but you don't hear it every day. And so when people that can look at the black and white and be skeptical are super excited, it's really hard as a parent not to be. So have a great night. Let's go to the splash pad. Everyone come with us to the splash pad, 6 to 8. We're going to see a lot of kids in water, which is their favorite thing in the world. We're going to play, we're going to eat, we're going to drink, and tomorrow we start again, 8:30.